Hello, my name is Ericka. I'm a master's student, and today I'm going to talk about the identification of PuuR as a transcription factor involved in Rhizobium phaseoli nodulation. First of all, Rhizobium phaseoli is a bacterium that fixes atmospheric nitrogen into the common bean plant. This is through the establishment of a symbiotic relationship in which there is a bidirectional communication between these two organisms. Now, the establishment of this symbiosis relationship is through flavonoids, which are metabolites produced by the plant, and that act as a signal in the bacteria through the transcription factor node D, leading to the transcription of nodulation genes, which is the first step of the symbiosis. But we don't know if there are other metabolites that can be mediating the symbiosis via transcription factors. Now, this is important because identifying other signals via transcription factors can aid in the rational design of biofertilizers. But in order to develop successful biofertilizers, we need to understand first what are the molecular mechanisms underlying the symbiosis process. Now, this is not trivial because we have a limiting factor in here, which is we don't know too much about the transcription factor gene regulatory interactions in Rhizobium phaseoli. Let me give you a perspective of this. We know that there are other 7 transcription factors involved in symbiosis besides NOD, but when we compare this number with the number of transcription factors known in E. coli, which is among 300, then we realize that we don't know too much about the regulatory mechanisms in Rhizobium phaseoli. So we ask ourselves, why do we use the available knowledge in E. coli to expand our knowledge in Rhizobium phaseoli? So the first thing that we did was to identify conserved transcription factors between E. coli and Rhizobium phaseoli. We did this by employing an approach called bidirectional best hit, which is an approach based on sequence similarity. And because of the divergence time of these two organisms, we use protein sequence. And the image each dot represents a transcription factor, and we identify 57 conserved transcription factors between these bacteria. After we identify the conserved transcription factors, we ask ourselves if the DNA binding motif of the transcription factors were conserved. This motif is the one that recognizes DNA sequences. And if the motifs were conserved, we assume that they recognize the same DNA sequence, and then we could predict the regulated genes of the transcription factors. We did this by employing an approach of 3 steps. First, we retrieve all the strip sequences of the Rhizobium phaseolis genes. Then we download these kind of matrices from Regulon DB, which is a highly curated database for E. coli. These matrices contain the information of the DNA sequence recognized by the transcription factor. Then we scan the observed sequences with these matrices. And finally, by scanning these, we identify the genes that are regulated by the transcription factors. We ended up with a regulatory network comprising 16 transcription factors colored in blue and 456 regulated genes colored in gray. Now, the next question that we asked ourselves was, are there any transcription factors regulating symbiosis-related genes? To answer this question, what we did was we grew up Rhizobium phaseoli with and without root exudates, and then we performed an RNA-seq analysis to finally identify differentially expressed genes. And gathering all this information, we identified 2 genes that were differentially expressed, that were also part of our regulatory network, and that were previously reported as symbiosis-related genes. Among these genes was not I, that actually it was regulated by a PuuR ortholog in Rhizobium phaseoli. And what caught our attention was that PuuR in E. coli is known to bind putrescine, which is a polyamine. And that led us hypothesize that maybe putrescine might be also binding to the PuuR ortholog in Rhizobium phaseoli. To test our hypothesis, what we did was we predict the tridimensional structure of these 2 proteins by using AlphaFold 2. And then what we did was to align these tridimensional structures to see whether the folding pattern was conserved or not. We actually found that the putrescine binding domain folding pattern was conserved. But we wanted to go a step further, and what we did was to identify the amino acids that are in direct contact with the putrescine, and we identified that six out of nine amino acids, which are in red, are conserved, while 2 out of 9, which are in orange, conserve their biochemical properties. These results suggest that putrescine binding domain is highly conserved. Finally, we ended up with a model in which putrescine plays a role in the symbiosis via PuuR. And this is achieved by the following steps. First, PuuR transcription factor binds to the promoter of the nodI gene, leading to its transcription. This nodI gene codifies for a secretory system, which allows the export of some signals from the bacteria that signals root cells, triggering nodulation. And once nodulation occurred, the plant produces putrescine, which is then internalized by the bacteria. And once internalized in the bacteria, it interacts with the PuuR transcription factor, causing its unbinding of the promoter of nodI, and this leads to eventually to the repression of this gene. Coupled with other processes, this leads to the second phase of the symbiosis, which is nitrogen fixation. Finally, I want to thank all the people who make this possible. Also, the funding agencies in Mexico, which are CONACYT and PAPIT. And I will be glad to answer any questions if they are. And thank you for your attention.